Today, I'd like to talk about human diversity. Let's begin with asking, what is a species? A species is a population or group of populations that is capable of interbreeding and is reproductively isolated from other populations. The members of a species can produce viable offspring that can live and reproduce. Given this definition, I should not have called Homo neanderthalensis a different species, but better, Homo sapiens neanderthalensis. How does speciation occur? Basically, when gene flow is interrupted and genetically isolated subgroups are maintained, new species may arise if the subgroups become incapable of interbreeding. I've already presented two models for the rate of speciation. Gradualism says that new species arise gradually over time through accumulation of small modifications. Whereas punctuated equilibrium states that a species remains stable, that is, in equilibrium or oscillating, for a long period of time, punctuated by sudden changes or evolutionary leaps. Think how the fossil record that we've just talked about reflects both of these models. In gradualism, among the fossil record, you expect to see transitional forms between species. And where did we see a lot of transitional forms? Well, between Homo erectus and Homo sapiens, and these transitional forms are grouped together as archaic Homo. Whereas with punctuated equilibrium, where we might have mutation of large numbers of genes, or mutations of genes that code for important phenotypic traits, and when combined with founder effect, can result in a new species fairly quickly. So a burst of change forms a speciation event, when a portion of a species is physically or genetically isolated. And so we expect not to see transitional forms between species, but suddenly a new form. So where don't we see transitional forms in the fossil record? Well, between quadrupedalism and bipedalism. And another place is the beginning of Homo erectus. Let's talk now about variation in species. Some species are more variable than others. How variable depends upon the species' geographic distribution and the variety of environments to which its members are adapted. That is, are they adapted to just a narrow niche or to a number of niches? If the species is adapted to a narrow niche, the species will be fairly homogeneous. That is, it will be a specialized species. An example of this is the koala. Living in Australia, the koala eat only eucalyptus leaves. If the species is adapted to a wide geographic range and many niches, it is a generalized species. Examples of this include humans, dogs, and grizzly bears. Generalized species are probably polymorphic. Poly meaning more than one, morphic, shape. So species with alternative alleles or species with variation, a polymorphic species. So polymorphisms are traits that display variation within a species. And if you're living in a variety of environments, you will need polymorphisms. Polymorphisms may show discontinuous variation. That is, the phenotypes fall in discrete groups. And if so, we can characterize these in percentages. An example would be blood types. When we look at the world population weighted mean of blood types, 41% of the world's population has blood type O, 32% blood type A, 22% blood type B, and only 5.5% blood type AB. Whereas a polymorphism may have continuous variation in which the phenotypes show a smooth gradient between two extremes. This then usually makes a bell-shaped curve when you're illustrating the gradient, and generally involves polygenic traits. And this may be described in terms of the mean. Examples of continuous variation in a polymorphism include stature, skin color, weight, and intelligence. And let's talk for a minute about stature or height. And although height is one of the most heritable human traits, cross-population differences are believed to be related to non-genetic environmental factors. And of these, fetal growth, which itself is related to maternal size, nutrition, and environmental exposures, and nutrition and infections during childhood and adolescence are particularly important determinants of heights during adulthood. So, information on height and its trends can therefore help understand the health impacts of childhood and adolescent nutrition and environment, and of their social, economic, and political determinants on both non-communicable diseases and on neonatal health and survival in the next generation. A recent study looked at the stature of men 1 century apart. So they looked at the 18-year-old men who were the age 18 in 1914, meaning they were born in 1896, and compared it to the cohort of men who were 18 in 2014, 100 years later. And looking at the top map first, you can see that North America had the tallest men in the world, as well as a little bit of Scandinavia. And if you look at the bottom chart, well, also going back to the top chart, you can see that the smallest mean height was over in Indonesia and Laos, where 18-year-old men were the height of what would be considered a well-nourished 12-and-a-half-year-old boy in North America. Now, if we look at the bottom map, a very interesting and unexpected trend. First of all, the mean height is now higher than it had been 100 years before. And you can see that American men did not gain substantially in height, whereas men in Europe became substantially taller. So America then, men did not gain as much in height over that 100-year period as did many other countries. So for height, genetics determines a range and environment determines where people end up within it. In other words, each of us has a genetic potential height. So if the nutrients you get in early life aren't enough, you don't reach that potential. And the authors of this study say that the U.S. has fallen behind because of the delivery of medical care, and a large segment of the population is uninsured, and the population doesn't get good nutrition and health care early in life. They pointed out that the U.S. has the greatest poverty rate in the developed countries. About 20% of American children grow up in poverty. And interestingly, the authors then of this study argue that height is a better reflection of the quality of life in a country than is gross national product. Let's move on to talk about the causes of variation in humans. There are 4 primary causes. One is sexual reproduction, which results in variation in individuals through mutation, recombination, linked genes, and segregation during sexual reproduction. A second are the evolutionary causes that we see variation among or within populations due to mutation, natural selection, gene flow, and genetic drift. And we talked about both sexual reproduction and evolutionary causes for variation in humans early in the semester. Today, I'd like to talk a little bit more about causes based on environment and on culture. Before we progress, just a word here about natural selection, which should be viewed as a conservative force, eliminating what doesn't work adaptively and allowing to reproduce what does work adaptively. So long as the environment or other selective forces remain the same, there's no change in what does or does not work adaptively. Turning to environmental causes, humans have lived in all kinds of environments for about 6 to 8 million years, and modern humans for 300,000 years. As an example, anthropologists and physiologists alike have sought to determine if there has been genetic adaptation to high altitude, which is conventionally defined as above 8,250 feet, since that is where the O2 saturation levels in the arterial blood begin to fall in most persons. And indeed, they found that among Andean populations in Peru and Bolivia, as well as populations in Tibet, we do see some genetic differences that have evolved based on their specific environmental conditions. And we do see greater efficiency of O2 transfer and utilization. Another case are the Bajau of the Philippines, Indonesia, and Malaysia, who can dive to 200 feet deep and hold their breath underwater for 13 minutes. How can they do this? They have evolved a larger spleen, which is an organ that supports your immune system and recycles red blood cells. And it's known that spleens of marine mammals that spend a lot of time underwater are also large. They also have a specific gene, which in mice is linked to spleen size. So here is yet another example of environmental conditions and way of living that have led to changes in the gene pool of this specific population. Sometimes humans have geographic barriers to gene flow. Examples that we've already talked about include Homo floresiensis and Homo luzonensis on islands, which were cut off by rising seas when glaciation ended. And their changes are linked to both natural selection, gene flow, as well as genetic drift. But humans may have cultural or social barriers to gene flow. Endogamy is a social rule in which you must find mates only within your own group, whether that's a cultural group, religious group, or ethnic group. Exogamy is a social rule that you should find mates only from outside your own group. However, with humans, it can be difficult to tell whether a polymorphism is or was adaptive in terms of natural selection, or whether it is random. Traits that were formerly disadvantageous, like hearing defects or certain chemical imbalances, may now be passed on to future generations because our society can correct some physical impairments. As an example, why do the Pima of the Sonoran Desert, located in southern Arizona and northern Mexico, now have the highest documented rate of diabetes in the world? And this is a problem that other long-time desert-dwelling groups have. They are also prone to high rates of diabetes. Up to 35% of the Pima developed type 2 non-insulin-dependent diabetes mellitus by adulthood. And their diabetes-related mortality is 10.8 times higher than among Caucasian Americans. Even horses like this Puerto Rican Paso Fino, his name is Cisco, after many generations in a harsh environment on an island, this breed is now prone to diabetes. Well, not really diabetes, but kind of. And for Cisco to remain alive here in South Carolina, he had to wear a muzzle every day so that he could not ingest too much sugar in the grass. Let's return to the Pima of the Sonoran Desert. Their problem has existed only since World War II. Prior to then, they had no diabetes. Why? What happened in World War II? Well, the Indian Health Services looked at their traditional diet compared to their diet since World War II, and they found that the Pima were eating the same amount of carbohydrates. So they told the Pima it was a hereditary problem. They could do nothing about it. They had a thrifty gene. The thrifty gene hypothesis was first put forth by Dr. James Neal in 1962. And he suggested that populations whose ancestral environments were characterized by periods of feast and famine experience positive selection for alleles that promote the storage of fat and energy. In other words, they selected for very efficient bodies. Moreover, under modern conditions, populations with such thrifty metabolisms are expected to have high rates of type 2 diabetes mellitus and related traits such as obesity. So, researchers took a closer look and they found that a mixture of genetic predisposition, cultural practices, and environmental factors played a role in the advent of this diabetes since World War II. Let's look at the environment first. The Pima live in the Sonoran Desert, and plants that grow in the desert are different from those that grow elsewhere in temperate or tropical areas. Some, like this Cholla cactus, produce a lot of mucilage. Some produce a lot of fiber, and some have slow-release carbohydrates. And the Pima became adapted to plant food with these qualities, never developing resistance against diabetes. So the desert plants, when eaten, do not cause blood glucose surges. So their population did develop a genetic predisposition for diabetes, one that was latent or not in effect with the traditional diet. Their cultural practices contributed to this also. Traditionally, when food was available, they ate a lot, and when there was no food, they went hungry. So their feast-famine traditional eating schedule did not select against diabetic tendencies. And their traditional ways of fixing the food, for example, grinding by hand, which results in larger particles that you digest, pit roasting, these both help slow digestion and prevent blood glucose surges. Why World War II? Well, the Pima changed their diet when Indian servicemen were exposed to Western food during World War II. That is, they joined the army or the armed forces and went off to war. And the carbohydrates in their new diet are fast-release carbohydrates, creating surges in blood glucose. You've probably experienced these. You eat lunch and about half an hour later, you're a little bit sleepy. Now, even when told their traditional diet is critical to their health, they don't want to return to it. They now prefer Western food. In other words, alleles that were once adaptive in their feast and famine situation in the desert may become maladaptive if the situation changes. And alleles that were once maladaptive may lose their disadvantage if the environment shifts. And culture may render some neutral. So what patterns of human variation do we see? Today, we accept the Clinal model. This says that genetically inherited traits usually change gradually in frequency from one geographic area to the next. This is the pattern we see in blood types, for example. We see a link between human blood types and certain infectious diseases because the two co-evolved together. Blood is typed according to the protein and sugar compounds on the surface of red blood cells. And some microbes have substances on their surfaces similar to these compounds, and thus they can trick a host with that blood type into not producing antibodies to fight off that microbe. The O allele is the most common blood type worldwide. People with type O blood are more susceptible to bubonic plague and cholera, and less susceptible to syphilis. The A allele is most common in Europe. People with A-type blood are more susceptible to smallpox. The B allele is most common in Asia. The smallpox virus is a mutation from one of the pox viruses affecting domesticated animals, such as cows, sheep, goats, horses, and pigs. And only since humans domesticated these animals have humans gotten smallpox. We've also seen a link between blood type and non-infectious diseases. For example, people with blood type O are prone to ulcers, whereas those with blood type A might be a little bit more susceptible to stomach and cervical cancer, ovarian tumors, and dangerous blood clots. Recent research indicates that the A, B, O blood types evolved at least 20 million years ago in an ancestor common to humans and other primates. Who among you cannot digest milk or gets gas or bloating if you ingest milk or milk products? I'm one of those. So is it normal or is it unusual to not digest milk? This is kind of a big issue in America because milk is pushed as one of the nutritional foods that you should eat. Mammalian milk contains complex sugar called lactose. For adults to digest milk, their bodies need to produce an enzyme called lactase. Lactase breaks down the lactose into the simple sugars of glucose and galactose, which can be absorbed into the bloodstream through the walls of the small intestine. In all human populations, infants and young children can digest milk. And in most mammals, however, including humans, the gene coding for lactase production switches off by adolescence. Intestinal bacteria can buffer the effects, and these bacteria increase with previous exposure, so you can acquire some tolerance for digesting milk. Adults who are lactase deficient, as shown in the picture on the right, when they drink milk, the lactose in the milk accumulates in their large intestine and it ferments, producing gas, and their gut swells with water and they get diarrhea. It's very uncomfortable. Most adults in the world cannot digest milk. They are intolerant. The Bantus and Ibus, some tribes in Africa, are almost 99% intolerant, as are Thais, U.S. Orientals. Australian Aborigines, 85% of them cannot drink milk, and 70 to 77% of U.S. blacks. When you look at the adult worldwide lactose intolerance, up to 70% of the world's population cannot digest milk. So it's America that is strange. Now, some populations are less intolerant. And why? For example, all but 4% of the Swedish can digest milk, and only 4% are intolerant. Only 12% of the Swiss cannot digest milk. Anywhere from 2 to 19% of U.S. whites cannot digest milk. And the Fulani, a tribe in West Africa, all but 22% can digest milk. Why this one tribe in Africa and not the others? Well, tolerance appears related to a long history of cultural dependence on milk products or pastoralism of cows or goats. Here you see a Fulani woman milking a cow, and the Fulani are famous for their cow herds. Let's move on to asking how close we are genetically to other primates. Back in the 1960s at UC Berkeley, researchers looked at overall immunological reactions of blood proteins from different species. That is, they looked for the antigenic distance between different creatures, humans and other primates, for example. So they looked at living organisms to quantify the similarities and differences in the immunological reactions of blood proteins. And if they're alike, then the number is 1. And so humans compared to humans, 1, they're alike. But humans compared to cows, 20, very different. Humans compared to a loris, which is a type of prosimian, 11.2, so not very alike. Humans compared to Old World monkeys, 2.38. Humans compared to orangutans, 2. But when you look at humans compared to gorillas and chimpanzees, we are all very similar. They hypothesized that this indicates not only evolutionary distance, but also evolutionary divergence. And in fact, you could use the differences as a type of molecular clock. given that these mutations causing these differences take place at a fairly constant rate, which is true only when you're looking at neutral mutations. So this was prior to the fossil evidence that we found more recently. They estimated back in the 60s that humans and chimpanzees shared a common ancestor about 5 million years ago. And they appeared not to have been that far off in the grand scheme of things. So molecular clocks work on the concept that over the course of millions of years, mutations may build up in any given stretch of DNA at a reliable rate. And if this rate is reliable, the gene can be used as a molecular clock. Now, using a molecular clock to estimate divergence dates, that is, for example, when chimpanzees and human lineages split, depends on other methods of dating. So in order to calculate the rate at which a stretch of DNA changes, biologists must use dates estimated from other relative and absolute dating techniques. If you look at this cladistic tree, you will see humans and chimpanzees are the most closely related. Following that are gorillas. Following that, our next closest relative would be orangutans, and then the lesser apes. So this cladistic tree was also a question on your test that you had this week, and many of you put lesser apes at very odd places. So obviously lesser apes are less related to humans than are the great apes. And among the great apes, gorillas and chimpanzees are the most closely related, and really humans and chimpanzees are very similar genetically. And lorises you see clear way over on the left among the prosimians. Pretty long time and far away before we shared a common ancestor with them. Researchers have also looked at amino acid sequences in certain blood proteins, and this is called sequencing. And looking at the percent difference between human and loris, clear over on the left among the prosimians, quite a lot of difference, 11.4% different. Humans and old world monkeys, 3.9%. Humans and orangutans, and remember, we've been separated for at least 13 million years, 2.8% difference. Humans and gorillas, 0.6% difference, and humans and chimpanzees, 0.3% difference. In DNA hybridization, you compare the actual genetic material, and this is based on the ability of one DNA strand to bond to its homologous pair. So remember, we have pairs of DNA strands, and if you were to separate those pairs, having just one of them, and you mix it with one of the homologous pair of DNA from another species, then you see how well one kind of sticks to another. And you can determine the tightness of the new bond by measuring how much heat it takes to break the bond again. And the lower the number, the better the bond. That is, the more alike the DNA is between those two species. And when you compare humans to loris, again, quite different. Old world monkeys, also different. Orangutans, different. But you get down to gorillas and especially chimpanzees, fairly similar. So humans and chimps are more similar than a horse is to a zebra, or a sheep is to a goat. Another way of looking at differences are chromosome band patterns. So if you put dye in the chromosomes, you can compare and see that among humans, chimps, gorillas, and orangutans, we have 98% similarities in our chromosome bands. They observed 500 bands. One of the biggest differences they noticed is that humans have 46 chromosomes, that is, in 23 pairs, whereas great apes have 48 chromosomes. So our chromosome number 2 is derived from two of the apes' chromosomes, now called 2A and 2B. So losing chunks of DNA is perhaps what distinguishes humans from other primates and has led to us developing bigger brains and other distinctive traits. We lack at least 510 chunks of DNA that chimps have, but only one gene. The rest may be enhancers, important switches for turning on genes during development. So researchers calculate that the genome-wide nucleotide divergence between human and chimpanzee to be just 1.23%. To summarize, a species is a population or group of populations that is capable of interbreeding and is reproductively isolated from other populations. Speciation can occur when gene flow is interrupted and genetic isolation is maintained if the subgroups become incapable of interbreeding. The two models for the rate of speciation are gradualism and punctuated equilibrium, and we see both of these in the fossil record. A species can be specialized or generalized. If they are specialized, they are adapted to just a narrow environmental niche. If they are generalized, they're adapted to many niches. Generalized species are probably polymorphic, and polymorphic traits can occur discontinuously or continuously. If they are discontinuous, we can characterize them by bar charts and percent. If they are continuous traits, we can characterize them by bell curves and averages. Human cultural practices can lead to separation of populations through practices such as endogamy or exogamy, yet gene flow is the overall rule among humans. Human culture can also mute the effects of natural selection or environment. Cloning refers to making an exact genetic copy of a creature. Genetic enhancement refers to adding genes to produce desired traits. Genetically, we are quite close to the great apes, and particularly close genetically to the chimpanzee. In fact, based on comparison of the human genome to the chimpanzee genome, only a small subset of the observed gene differences is likely to be responsible for the key phenotypic changes in morphology, physiology, and behavioral complexity between humans and chimpanzees. Can we say what these are yet? Not quite. We need to figure out a whole bunch more stuff and look at more information and evidence, even though we have the genomes of both. But among ourselves, humans are 99.9% genetically identical to each other. Chimpanzees have 2 to 3 times more variation within themselves than we do among ourselves. So think about that. Humans are spread all over the world in so many different environments, whereas chimpanzees have a much more limited geographic spread, and yet they have 2 to 3 times more genetic variation than we do.